Okay, I think we can get started now. Uh, so thank you everyone for joining us today. We are very excited to host one more webinar in this WNAR webinar series. First of all, I'd like to let everyone know that the session is being recorded so that we can share the video afterwards on the WNAR website. If you have questions, please put them in the Zoom chat box, and we will handle all the questions at the end during our Q&A session. So it's my pleasure to welcome Dr. Daniel Fink today. Dr. Fink is a senior research associate at the Cornell Lab of Ornithology. He received his PhD in statistics at Cornell University in 2002. And for many years, he has been conducting his research on statistical modeling, machine learning in a spatiotemporal context to solve challenging problems in ecology and conservation. We are very excited to have him with us today, and we will look forward to hear him present on some of his work on modeling some eBird data. And with that, I will turn this over to Dr. Fink.
Thank you, Pracha, and thank you very much for the invitation to speak today. I'm glad this is working out. I have a number of slides. I guess if we get towards the 45-minute mark, please feel free to rein me in if I keep going. So what I want to do today is, I'm assuming that most people aren't very familiar with eBird as a project, and so I wanted to provide an overview of the Status and Trends project. This is a project I've been connected to for a number of years now, and it's it is basically an applied statistics, a big applied statistics problem, and so much of my work has been in various aspects. What I think about is somewhere between research and applied statistics. And what we're trying to do is we're trying to extract useful biodiversity information from this large community science project. So with that, let me get into it a little bit. I guess I wanted to start. There we go. I wanted to start with acknowledgements just to make sure that they get in. So I'm working at the Lab of Ornithology. It's a large organization that's connected to Cornell University. The lab is kind of considers itself almost like an NGO. It's a science-based and kind of research-forward organization, but we also have members that join, and a wide variety of different programs that do things from conservation science research, or more traditional ornithological research or ecology, all the way to more recently, more data science kind of focused research, trying to tackle interesting problems related to various data streams that the lab's been collecting. I mean, so it's kind of a wide group. The group that's, there's a large number of people that are attached to the project and should be acknowledged for this work. This top row consists of kind of the core group of people that do this work. These are geographers, statisticians, data scientists and engineers, as well as kind of in the second row, our core group of ornithologists and really like expert natural historians that understand birds in the field in a way that's quite amazing. The other thing is I should note that the data that we're going to be talking about is contributed by volunteers, and it's a huge data set, and there's sort of millions of hours worth of thank yous involved, and we also have a number of organizations that help make this happen and have been funding this work. Okay, so with the preamble, what I want to do is kind of jump into the problem at a really high level. So why birds? Why a lab of ornithology? And sort of what is it that we do? birds are easily detectable. They're almost everywhere on the face of the earth, and people just love them. And people have for a long time who've been looking to the skies, dreaming about flight, or just enjoying birds for the sake of birds. And there's a long tradition of people keeping records of what they see. So there's a huge kind of human engagement component. Birds are surprisingly intelligent and adaptable. I can say that because my pet bird, our rescue bird, has a mind of his own and a personality to match. And what this makes them is very sensitive indicators of environmental health and environmental change. So from a biodiversity perspective, birds represent one of the most information-rich taxa on the planet. And there's this huge love affair going on with them. So there's a way to tap into this and collect a lot of information. Despite this, and despite the sort of history of people watching birds, it's still hard to collect enough information to be able to understand some like basic ecology and sort of basic information for biodiversity management on birds. If we think about the movement of these populations across continents or across hemispheres, and to try to understand that really at fine temporal and spatial resolutions, the information kind of requirements are daunting. And so this is where the project eBird comes in. People at the lab, probably 25 years ago or so, had this idea as the internet was kind of coming into being that it would be possible to harness these energies and motivate people to not only watch birds and to help them collect and collate and organize their information about them, but to also be able to harness this information for science, for biodiversity studies and conservation. And so eBird started in 2002, and eBird is a bird monitoring project. It engages a global bird watching community now, and people submit their observations of birds through a free mobile app and through a website. There's a part of eBird, the project, is a large effort to engage different groups of naturalists and different kind of audiences at different places in the world to engage people to identify and record the birds they see. So this has benefits for environmental education and sensitivity. It also generates a huge amount of data. The observations that people collect are organized as a checklist of species. And so the picture you see here is a picture of the mobile app. There's a lot of design that's gone into the app to make it easy for people to submit this checklist that identifies which species they have detected and identified. And to be able to do this efficiently, you can imagine if you have potentially hundreds of species to sort through, this is, it can be cumbersome, and to do it well may require a fair amount of effort and expertise. So the idea is for the project is kind of to lower that bar as much as possible, but to maintain some kind of enough integrity of data that we can do valuable things with it. So the project has been really successful. This map shows a fairly recent snapshot of the locations where eBird checklists have been submitted. And I mean, there's a huge number of them, over 10,000 species. I'm reading some stats, over 60 million checklists. We're kind of approaching close to a billion individual birds that have been counted. Data comes in now at roughly, I think it's over 1 million observations per day. So it's just there's a huge audience. However, when you look at this map, you can also see there's some large gaps. There's still a lot of bird data collecting that's not happening. And so that presents some interesting challenges as well. I'm gonna focus mostly on the Western Hemisphere and North America for these examples. So this is where the project has originated, and the sort of outreach to the communities has been has the longest efforts in the US and North America. And so data density here is the greatest. The data set that we work with on this project, the Status and Trends project, consists now of about 26 million checklists at 6.5 million locations through the Western Hemisphere. And what's unique about this data set is its size and scope, both spatially and temporally. Data is collected year-round from any location, and that's different than data. Other kinds of taxonomic data have been collected, and bird data kind of up until very recently. What's really exciting is this amount of data, and so the challenge, is what we can actually do with it. So with this kind of comprehensive and detailed information, the particular things that we want to estimate, some of the basic biodiversity information about species are the range, where they occur, or sometimes called distribution, the abundance or density of the population in an area, associations with habitat, and then year-to-year variation or trends in population size. This is the basic information that helps drive conservation planning across a range of scales, and also ecological studies. So to be able to deal with eBird data, this is data that comes from this community science project. It's basically, you think about this as a large crowdsourcing project in terms of the way data is collected. There are a number of challenges that arise. So the first one is the ecological processes. These are the processes that connect species to particular places at a particular time. And these can be complicated. There's very often multiple simultaneous processes at play. You can imagine this is things like climate, topography, land cover, land use. This is all often at play at the same time. The observation process by which our eBirders go out and collect these checklists is also complicated. And like other large scale community science or crowdsourcing projects, the data that we end up with are imperfect and incomplete. And then there's the scaling challenge. So both of these processes, the ecological and observation, vary strongly regionally across space and seasonally throughout the year. And this creates kind of like more challenges. So the framework that we use, the high level kind of methodological framework is the framework of species distribution models. These go by a variety of different names within the field or fields, sort of ecological domains. These are also known as niche models. And the idea is to connect the observations of species and link those with environmental data and observation process data. So at a really high level, there's two goals here. One is to try to fill gaps in space and time, where these are basically unsampled locations. And the second one is to remove sources of bias associated with the observation process. And so if we have reasonably good models, then we should be able to extract some useful ecological information. I guess the one thing to say for this audience is I think of this framework as essentially a high dimensional regression framework. And so statistical learning, machine learning plays a role here as well. And I'll tell you a little bit about that as we get further in. So first, what I want to do is I want to give you an idea of like what kind of information it is that we can extract from eBird. So we're going to start with this animation. This animation shows estimates of the relative abundance for wood thrush. This is a migratory bird or a neotropical migrant that winters in the Yucatan Peninsula in Central America. You just saw it kind of cross the Gulf, and then it spends its breeding season or summer in the eastern North American deciduous forest. In the visualization, lighter colors are areas with lower relative abundance, and darker areas are where the population is more concentrated. The estimates that you see here are made at 2.8 kilometers at a weekly time interval. The gray areas, there's two different colors of gray you'll see. The dark gray are areas where there's enough information to make a prediction of either positive abundance or a zero. That is where we're making a prediction the species does not occur. The gray areas are where there isn't enough information to do that on any given week. And so you'll sort of see that boundary dance around a little bit, reflecting the varying data density underneath. What's new and interesting about this is, one is the temporal resolution. The ability to do this throughout the year is something that was unique with eBird. And the ability to also do it at spatial scales that are this broad and to maintain some moderate amount of spatial resolution, the 2.8 kilometer resolution. So to provide this information is really valuable for a number of different applications. I mean, this is a way you can think of, it's kind of a first way to visualize where these populations are and how they move. It provides really basic information of like where the population is and when it's there. This is critical for conservation applications. For ecological and conservation audiences, this information is distilled in a number of ways. And this is something we do as part of the project and kind of the data products that we make available. There are seasonal summaries. So here, like the blue is the winter or non-breeding season. Red is the summer or breeding season. You can see some yellow in between. These would indicate some of the migratory areas that are, or areas that are where the species occurs only during one of the migrations. So you can start to see, like, there's the ability to do things like be able to track for a given species where and when the population is concentrating. This kind of information, there's a large audience for this information, even when it's kind of coarsened down to binary information for each of the seasons. So this is almost like a range boundary for each of the seasons. As part of the modeling process that goes on, we also quantify the information about the habitat associations or land cover associations that drive these predictions. This is an example to kind of give you a feel for what that looks like. So again, this would be wood thrush. We're looking at a map of its distribution during the breeding season. This green square here, if we focus just on this area, we have summarized the associations or predictor importances with wood thrush occurrence and a variety of different land cover types that are used in the modeling process. So just to give you a feel for what the information here, if you look at the x-axis, we're going by month across the year. The y-axis is the relative importance of the predictors. Anything above 0 reflects a habitat that is used by the species, and those that are below 0 are habitats that are avoided by the species. The colors indicate each one of these different land cover types. You can think about these maybe loosely as habitats. And the thickness of them at any given time represents the relative importance of them. So it's interesting, if you look in this area, what you can see is there's no importance up until early March and then into April. This is when the wood thrush are migrating into this area. There's a relatively large number of different colors if you kind of cut through, take a vertical slice through here. This represents this is the sort of more variable or diverse set of associations as the species is actually in during migration. During breeding season, we see this green area is really this wide, representing a really strong association with deciduous broadleaf forest. And then into the fall, what's interesting is sort of as the population is starting to move, we start to see a more diverse set of colors. The association with the deciduous broadleaf drops off. But what's interesting is that the association with urban built areas increases. The modeling process, and I'll show you a little bit about this, is allowed to vary in space and time. It allows estimates of these associations that also vary in space and time. So if we look at this region that includes Atlanta, here you can see that this seasonal habitat association plot shows a different pattern. We still see this like strong kind of bulge of the deciduous broadleaf forest during the breeding season. But what we can see is there's an avoidance of some other different cover types. These are ones that are more prevalent in this region. But we also see, notably, this large increase in positive association with urban areas in the fall. And when we started doing this several years ago, we saw, one thing that was interesting is we saw with a number of these migrating species, these associations with urban areas. And for the longest time, we thought these were errors because these are species that are known not to like urban areas. And so we would see a little bit in the spring and a lot in the fall, these strong associations. Since these sort of early days, colleagues have actually run experiments, and there's at least one process that we know of now that actually attracts birds, these migrating birds, especially these nocturnally migrating birds into urban areas, and that's night lights. There have been some interesting experiments done turning on and off strong urban lights and recording changes in nocturnally migrating bird densities that line up with these kinds of analyses. So it's kind of an interesting side story. Similarly, we can quantify relationships and more of the actual shape of the relationships between some of these different land covers. This is an example with ring-necked duck in January in New England. In this plot here, these different colors represent the different seasonal distributions of ring-necked duck. The blue here, these are the areas where the species is present in the winter. So this would be the January area. And this plot here shows a partial dependence plot on the x-axis showing the percent of a 3-kilometer neighborhood that's covered by lakes. And the y-axis shows the deviation in our logit occurrence. So if you look at the occurrence, it looks like ring-necked ducks will tend to occur when you have at least maybe 10 or so % of lakes covering a particular area in the winter. We can also look at this as a function of the counts of ring-necked ducks in the areas where they do occur. This is interesting because the peak looks like it's closer to 30%. And so these two pieces of information, this is valuable information for understanding some of the habitat requirements for the species, this particular region and this particular season. The last kind of category of information that we generate as part of the project are year-to-year trends. So what we looked at with the animation was the seasonal variation that's especially dramatic with migrants. The year-to-year variation here, this map shows the average annual percent change in abundance from 2007 to 2019. So decreases in population size are shown in red. You can see there's a little bit over here in the Pacific Northwest. Areas that are increasing are shown in blue, where the deeper the color represents a stronger trend. Each dot in this map is a 27-kilometer pixel, and the dot size is proportional to the population size. So the way this visualization emphasizes the change in the total population over this time period. And it's really interesting that most of the bald eagle population over this period has been increasing and increasing quite a bit. This is actually, it's a really interesting story because bald eagles have enjoyed a lot of conservation effort that's been coordinated at a national level for many decades. I believe since at least the 40s, the 1940s. So this is actually, behind this is actually a really interesting success story of national conservation. Okay, so I'm going to switch gears a little bit. That's a little bit like of an overview. I'm going to talk a little bit about methodology, and hopefully I'll have some time to get into some of this with you. I'm going to start with the data. The data set that's underneath all this. So on the eBird side, the observational data side starts with the species count. This is the count of each individual species that's reported by the user. The app or a person on the web records the when and where when a search took place. eBird also records, importantly, information about the observation process. So we've got a couple of key predictors that describe this. Search effort, how long participants spent in the field, how far they traveled, the time of day, the number of people in the party. So think about more eyes or ears might be better or worse for detecting species. There are different protocols whereby people search, and we have an index, which I'll talk about in a little bit. These are joined with information about the local environment. And so NASA remote sensing data is like one of the main data sources we work with and describe data at across the landscape at a 3-kilometer or 2.8 kilometer pixel. There's 21 different classes of vegetative cover, use, and hydrology. And then we join that together with nighttime lights, some information about islands and elevation. And this is described both in terms of how much of these land covers there is in the particular pixel, as well as how much variation there is within the pixel. So it's a fairly large, ecologically speaking, fairly large data set. I think we have in the realm of maybe 80 predictors. And that's enough information to describe populations across broad extents with this resolution. So to use this information for predicting species occurrence and abundance, we have a number of challenges. There's the ecological process, both occurrence and abundance. We have to deal with imperfect detection, variable search effort, the heterogeneity in how people search and how well they search, site selection biases, and technical problems dealing with lots of zeros and highly imbalanced data. At broad scales, we also have to deal with non-stationarity in these relationships and the density of the data, which you saw in some of those maps. So I want to start off by taking a look at, just giving you a feel for like what I think of as the base model that we work around. So this is what we're using is a 2-step hurdle model where we model the presence or absence, the occurrence of the species, and then conditional on that, the count. Both of these for both of these steps, we've been using machine learning models, boosted trees and random forest. They work fairly well, but they're tuned a bit for this particular application. Having the two steps in this way has proven to be pretty useful in terms of generating ecologically relevant information too. One thing we can do with both of these is we can actually look at the concentration of populations and how that changes. So in this plot, we have on the x-axis is a rank of the count part of that model, the number of robins that are detected, and in the y-axis is the probability of detecting one or more. So up here in this corner where it's really pink, this would represent if you had individual robins detected frequently. So for example, this is the distribution of, this is the map of American robins using this palette. What you'll see is that there are a lot of pink areas here. This is breeding season in June, where it's really pink. So this is during breeding season, the species spreads out as they breed in a lot of these areas, taking advantage of the area and the resources. The blue here represents robins that are detected infrequently, but in really large flocks. So if we look in the winter, what you'll see, so this is at the end of December, populations moved south a bit, and it's much bluer with some of these blue areas. And then if we kind of look week to week, what we can see is the shift from the winter where it's sort of light blue to dark blue and then pink. So with these the two submodels together are actually giving us some biologically meaningful information about how these populations are actually concentrating and spreading out throughout the year with as part of their annual life cycle. This also is invaluable information in terms of understanding where and when these populations are and how they're using their habitat. So there's lots of conservation information here too. Let's skip that. Another thing we have to deal with is imperfect detection, and basically it's variation in the observation process. And I just want to give you an idea of sort of two levels at which we deal with this. Some of the covariates that are collected by eBird, we use those to capture variation in that observation process. So we train models that include, like in this plot here, search duration, the amount of time that participants spend searching. This plot shows how the occurrence of species changes. So as you imagine, when you spend more time searching, probability of detecting and identifying the species increases up to some point, and then there's sort of diminishing returns after that. This plot shows how for wood thrush, the probability of detecting and identifying a species changes throughout the day. So this is the hour of the day. Into the dawn chorus, the species is much more easy to detect as they start to sing and communicate with their friends. And then later in the day, they get quiet. And in the evening, again, they get a little bit louder. So this is another source of variation we think about as the availability for detection. We want the strategies to describe as many of these sources of variation as possible, and then to remove them when we make predictions about the ecological process. And this is done by identifying the values that maximize the probability of detection. So each individual observation process predictor, we want to maximize this and then have an estimate of during the optimal time of the day, an optimal search effort when we can see these species. Another really important source of variation is the heterogeneity among birders. So on the left, you can see this might be someone like my daughter or someone like me who knows very little about birding, how to identify species. These folks are people that actually compete for the, I guess, for the trophy and the glory of being some of the best bird trackers and identifiers within a particular region. And the pictures below kind of give you an idea that sometimes looking for birds is hard and there's imperfect detection. So how people choose to search and what their skill level is makes a big difference in terms of how much variation we see in that observation process. And one thing that we've done to address this as part of the project is kind of a feature engineering project. So this plot here shows you the length of a checklist, how long people go out searching. And here the y-axis is the number of species recorded. So as you spend more time, not only are you more likely to see an individual species, but your accumulation of species kind of follows this increasing curve. Now this might be like my curve here, and this would be the curve of someone who actually knows their birds. You can imagine if you're skilled and you understand natural history, you know where and when to search and how to identify the songs and sort of habits of birds, you're going to accumulate species much, much faster. And so what we've done is we've used this, we've modeled this relationship to predict the expected number of species on a given checklist as an index to capture some of this variation between observers and between their checklists. And this has really been a very powerful tool. This picture shows a standardized measure of this feature. We call it the checklist calibration index. It's standardized to be 0 across a group of birders within a fixed region and season. This is one of my colleagues. He happens to be a very good birder. So if you think about this as a normal scale, he's up here on the top tail most of the time. But sometimes when he goes out to search, his scores for those particular checklists is really low. The number of species that he actually detected is much less than you would expect across this group of birders. And it turns out these are times where, times where my colleague has actually traveled to new areas where he's been forced to learn new birds. So this is an indication of how sensitive this measure is and how much variation it soaks up. This is another colleague I work with who started birding relatively recently. And you can see as he's learned more about the natural history of the birds in our area, he's steadily gotten better. This is a curve of another colleague who has steadily decreased in their score. And this is indicative of some of our elderly birders. As you get older, your ability to hear high frequency sounds starts to diminish. And high frequency sounds are key to identifying large groups of singing birds, in particular ones that really only can be detected by song. And so this is, in some sense, the senescence of one of our bird sensors. He hates this plot. When we include this index, it makes a big difference in the distribution estimates. It soaks up a lot of variation, and when we adjust to make predictions as though we had a very skilled observer, what you can see is the difference without CCI and with fills in a lot of this range. And based on expert review, this is a much more accurate reflection of where these species are. The same goes for validation tests for this. Trying to think, there's, maybe I'll quickly go through one of these other challenges is the site selection bias. So the data in hand that we have, as you saw from those initial pictures, this is another one in the Northeast US. These are locations for checklist for the month of June, I think in 2013. And what you can see is that there's a very uneven distribution, geographic distribution of search effort. In fact, if you're familiar with this region, this map looks an awful lot like a map of human activity, both towns and cities as well as highways. There are areas that are convenient to go birding. And this kind of problem is really typical of any kind of geographic crowdsourcing. So the data we have in hand is an uneven sample of the area that we want to make inference about. What the target population we really want to make inference about is every region. And we would, so uniformly distributed, where let's say every pixel has equal importance. So with that sort of difference kind of sets up our site selection bias. And we can model this. So we've modeled this using a binary classifier to discriminate between where people go and information about the entire environment. And what you can see is that if you model the probability of visiting a particular site based on this data, we end up with a map like this, where the darker areas have lower probabilities of selection, and the ones that are whiter have a higher probability of selection. Again, you can see this really strong pattern of human activity and roads here. One simple fix for this is to sample the data more evenly when we train these models. And when we do this, if you were to look at the site selection probabilities with and without the sampling, you can see that the sampling makes a difference. It definitely whitens up this image for a large sort of number of pixels across this region. It's not perfect, but it is an improvement. And this is something that we commonly do. There's one other challenge I'll briefly talk about, and that's scaling these distribution models. When I've been talking about them, and some of these images are sort of regional and seasonal, when you think about the ecology of this and how this sort of stationarity challenge plays out. Uh-oh, this is going by itself. Yeah, I'm sorry, the slide's advancing by itself. Here, I'll leave it right here. If we start by looking at this area, which is sort of the Carolinas and the Piedmont, tree swallows, which are common. I'm sorry. They encounter environments that look like farms and fields, but tree swallows also occur all the way on the other side of the continent in California. And there they encounter very different habitats, like intense agriculture. So this species has to find its suitable habitat in very different contexts. Our predictive models need to do the same thing. And what we found is that a divide and recombine strategy helped make this process easier and improve performance. So what we're doing is we're creating an ensemble of lots of regional models. So the divide portion is to partition our study extent, like here, the Western Hemisphere, into a randomized set of regions. We subsample the training data within each of these, assume stationarity within each of these, and then train, predict, and estimate. And we do this over and over again so that each area is covered by ideally 100 of these independent, sort of locationally jittered regions. I call them space-time pixels or stixels. And then the idea is when we make a prediction, we treat it as an ensemble, and we average for a single location in time, we average across all 100 models or however many in the ensemble are making a prediction at this point. So we're really averaging across an ensemble of decision tree ensemble models. For example, this year, each of these would have 100 random forest models that we average across. And this allows us to scale up in space and time. So this is, this picture is kind of fun because this is a species that moves and migrates from parts as far south into South America, all the way up into the northern portion of the North American continent and then back again. And the environmental resources that it encounters along the way are very different. The context is very different. And so the model is well suited to capture those. This is kind of the funnest one. This is the barn swallow actually is a nearly globally distributed species. And so for fun, we've scaled this up. You'll notice, especially with Africa kind of front and center in this visualization, that there's a big hole here that again represents areas where there's just not enough data to even to make any kind of inference at all or drive these models. So this also gives an idea of where the what data density in those first couple of maps translates into in terms of our ability to model distributions in abundance. Okay, I've given you a bit of an overview of like some of the different products and some of the challenges involved. This information is being used for different conservation projects and ecological projects and climate changes as well. One I want to highlight, I think I'll have time. Maybe I'll just do one. We're working with U.S. Fish and Wildlife Service. They approached us and they're really interested in the spatial extent and detail of the information that we have on eagles. Fish and Wildlife Service has been in charge of managing golden and bald eagles since the 1940s. There's federal law that mandates this. And so they're interested in trying to improve the application of the policy that they use. This project began by looking at the non-breeding or winter distributions for bald eagle, shown here, and golden eagle, which is mostly a Western US species, shown here. And the goal was to try to develop a more flexible and efficient scheme for certifying new wind turbine facilities. So wind power development. The policies up to recently, I think possibly still today, require that any new wind development site needs to collect 2 years of data on the occurrence of both of these eagles. And the idea is that the Fish and Wildlife Service had was, with this kind of information, that they would be able to identify which areas were of low risk to both species and could be exempt from the 2-year data collection requirement. And conversely, which areas really have high risks for collisions for either or both of the species and should remain under that 2-year kind of data collection requirement. And so Fish and Wildlife Service has a number of regionally distributed data sets that are known to be fairly high quality. And they worked to validate those against the eBird based estimates, and then came up with this map, which represents the proposed low exposure areas. And the idea behind this then is to make that permitting process more efficient, really focus conservation efforts where they're most needed, and to allow for wind development and make that more efficient where it's possible to do so. Okay, my clock says I'm almost out of time, so I'm gonna skip ahead and kind of wrap up here. I guess there's lots and lots of other challenges. Some that we're working on that I haven't shared with you, others that are new and interesting. Accounting for species to species interactions is something that we do have some collaborations working on that, and that's an active area of research and ecology. There's lots of fascinating questions there. The methodology to be able to reliably estimate year-to-year trends when the observation process is also varying from year to year is an active area of research now. And being able to more fully explore the species-environment relationships, specifically ones with climate and extreme weather have been some of the things I've worked on, and with a number of colleagues. The ability to improve our estimation of or separation of the occurrence and or the detection, I should say the observation process and the ecological process, or specifically the detection process and the occupancy process, is also another area of research. If you're interested, we have lots and lots of information and results available on our website. And there are a number of resources available, including the eBird data that goes into this model is available publicly. These links will take you there. We have some R packages to make it easy to pick the data up and use them, and some training materials to help people get up to speed quickly. Questions?
Thank you so much, Daniel, for the great presentation. We can jump onto the question and answer session now. So if anyone has any question, please feel free to unmute yourself and ask your question.
Hello, I have a question about the breeding season kind of in the beginning. Are you modeling the start and end dates of migration?
Daniel, you're muted, I think.
I am. I hope I didn't cut out too long ago.
No, you're good. So did you get the question?
No, I'm sorry. I was dropped from the Zoom. If it could be repeated.
Yeah, I think the question was whether you were modeling the start and end date of the migration, but feel free to correct me if I'm wrong.
That's the question, yeah.
We aren't directly modeling the start and end date. For each week, we do model, we make an estimate of if the species occurs in that pixel. So we make the binary prediction, and that contains some information about the beginning and ending of migration.
So I have one question. You talked about the CCI, the index. Can you give us some idea about how you actually use it after you obtain that index? Are you using it to model it as some kind of uncertainty measure?
Well, that's a good question. So with the index, what we actually do with that, what we do is we compute it, I guess a little background, we compute it across that ensemble, and then regionally we standardize it so that we can compare it across regions. It goes into the occurrence submodel as a predictor. We found that we've experimented using it as to model the variance in the occurrence or the mean of the occurrence, and it's a much more powerful predictor of variation in the observation process in the mean. So we include it similar to the other observation process predictors. And then when we make estimates of occurrence, we do so for a skilled eBirder.
Okay.
Hey, Daniel, can I ask if you have some favorite tools for dealing with the massive data sets?
Is this Lisa?
It is Lisa. Hi, how are you?
Good. It's been a long time.
It sure has.
This is a funny way to meet again, but I'll
I
take
know.
tools. Yeah, we've do you want I could definitely tell you about different kinds of tools, but do you have a specific? There's
Well, the GIS data, the satellite data is large. I mean, you've got just thinking about North America, it's enormous, and so there are just technical difficulties managing that. And I'm just wondering if you, since you have experience, do you have recommendations for good ways to manage data like that?
that's a good question. Yeah, so I gave a very high level overview of more of the data science side of the project. The data, what I consider the data engineering side, is there's also lots to be said. So at a very very basic level, we actually are still using R to do that processing, but the data engineers that work on the project deploy that. We've deployed that on AWS, and we've done that in a number of different ways over the years. More recently, we're doing it on Exceed. So it's a highly parallelized process that's happening lots and lots of processors. We do it globally, so it is a big task, but we've actually been able to scale the R processing so far. The only part that I may be wrong about is GDAL. I think it's possible that we're using a different version of GDAL, but I could find out for you if you want.
Yeah, maybe we could explore this offline. I think they updated GDAL at some R update. I ran into that a while ago. Yeah, thanks. So I'll just send you an email and we can do this offline.
Sounds good.
Hi, Daniel. I saw on one of your last slides, you mentioned year-to-year kind of trends in this data is something you guys are moving forward with.
Yes.
I was just wondering if, say, we took a small subset of the data and tried modeling it across multiple years, what do you think the accuracy of those year-to-year estimates would be?
I guess the first thing to say is the accuracy of any of these estimates varies widely with region, season, and species. And, and that's a function of like data density, detectability for the species, prevalence of the species. Like there's a number of factors. So a one size fits all generic answer doesn't exist. But if you had a species that had fairly good data, the bald eagle's a good example, you can, so far, we've been able to actually, what I think is do a pretty good job estimating those. And part of that is based on validation against independent data sets. And the Eagle project, we've done a bit of that with the map I showed you. So like that increase in the Pacific Northwest is something that's corroborated with independent aerial surveys of eagle nests. And the increases in a number of other places are also have been corroborated by the same kinds of aerial surveys in independently run in different parts of the country. We've also invested heavily in simulations, and the simulations kind of across species suggest that with fairly good data, you can actually do a pretty good job. So I think there's a lot of potential.
Yeah, thanks so much. Those bald eagle maps you showed earlier were really, really interesting.
We've reached the end of the time for today, so I guess we'll conclude.
One more question.
You have one more question?
Yes. Just to Daniel, would he put his GitHub link into the chat window so we can copy the link? Thank you.
Sure, I can do that. I think for the, I can put, let me put, one is the web page has a couple of links. So I'll put, I will try to put that and the GitHub for you.
Somebody else is asking if we can get the slides.
Yeah, we are going to share the slides afterwards on
Okay.
WNAR website.
There we go.
Okay, great. So on behalf of WNAR, I would like to thank Dr. Fink again for the wonderful presentation. And I would also like to thank everyone for attending, and we hope to see you soon again. Thank you, and have a very nice day.
Thank you.